Hi everyone, my name is Cassandra Burdziak, and today I am delighted to share my work in the Donna Pear Lab at Memorial Sloan Kettering Cancer Center, jointly with Julia Zhao, titled scKINETICS, Inference of Regulatory Velocity with Single-Cell Transcriptomics Data. And so to start with a bit of background, nearly all biological systems, ranging from embryonic development to cancer progression, involve some dynamic process through which cells actively change their cell state. Using single-cell genomics, we are now able to take a snapshot of these sort of processes from any tissue in which cells are asynchronously progressing from earlier states to their eventual fates. Using this data, there are a number of pseudotime-based approaches which attempt to order cells along a one-dimensional axis from the most immature populations to the most mature populations, with the ultimate goal of reverse engineering mechanisms of the process based on gene expression patterns. Now, in order to motivate our approach, I want to look under the hood for a moment at what these pseudotime algorithms are actually doing. So when we collect single-cell data, the cells are intrinsically unordered, and so we cannot see patterns of expression across any relevant axis initially. And so most pseudotime approaches are simply trying to find a reordering of these cells to reveal smooth patterns of gene expression, typically through manifold learning approaches. Still, there is an inherent challenge with interpreting these results, as an equally plausible solution to any manifold learning problem would be to reverse the axis entirely. And so hence, we cannot know which are the immature populations and which are the endpoints of these processes without using some sort of prior knowledge. All of this is to say, while biological processes are generally directional, and that direction matters to our understanding of the system, manifolds in most cases are not. And so what can we do to extend beyond this one-dimensional manifold representation of the data? So ideally, we would like to learn a function that describes how does gene expression change over time. Of course, the key problem here is that time is generally unmeasured. And so perhaps the next best thing would be instead to learn a function that describes how future gene expression will be impacted based on the current expression state of the cells. So it turns out that this is actually easier to model directly, and that we can imbue some mechanistic knowledge to write down this function without the need for observed time. For example, what if I told you that I know gene A, in color green here, has a negative impact on the expression of another gene, gene C? So looking at this plot to the left, in that case, we would see when we have a high level of expression of gene A at a particular time point, we would expect to see a decrease in gene C at all subsequent time points. Hence, we're incorporating a notion of time without necessarily modeling it directly. So what I'm describing here is actually something like a differential equation, where we model the rate of change of a gene as a function of the expression of other genes. And in this example I've described here, that would look something like this, where the rate of change of gene C is negatively related to the current expression level of gene A. And so this defines a class of methods referred to as velocity, which consider the rate of change across all genes as a vector, which describes the likely future state of a given cell. So how do we do this in practice? Well, previously, most approaches consider RNA splicing, where unspliced RNA necessarily drives the rate of change of spliced RNA for the same gene species. And this implies these sorts of causal relationships between unspliced and spliced RNA, imposing these sorts of dynamics over time, where for each gene here, we see that an initial increase in the unspliced RNA in the solid line is followed by increases in the spliced component in the dotted line. This is modeled through dynamic equations, where in this case, each gene's rate of spliced expression is modeled as a function of its current unspliced counts. And so while this method has proven very useful in some contexts, it turns out that it doesn't seem to work universally, and this could be for a number of different reasons. For one, splicing is very difficult to detect in 3' biased single-cell RNA-seq data, and some processes don't seem to be driven by genes which are dynamically spliced in the first place. And so our simple idea to solve some of these issues is just to replace this model with a different notion of causality, in this case, which transcription factors, or TFs, causally influence the rate of expression of some of their downstream target genes, also imposing similar causal relationships and corresponding dynamics. This is instead governed by a differential equation per target gene, whose rate is impacted by the expression of its regulatory TFs at the current time point. And so with this in mind, we developed an approach which we are calling scKINETICS, Key Regulatory Interaction Network for Inferring Cell Speed. And it has this basic idea where the expression of each gene I is impacted by the current expression level of each of gene I's regulatory TFs, which in this case is denoted as J. And importantly, because gene I itself can be a regulatory protein, we're able to model these types of regulatory cascades where each gene's expression is impacted by the dynamics of the entire upstream gene regulatory network. As stated, we do this with a system of differential equations consisting of one equation per target gene, with rates of expression dependent on both the transcription factor's expression and this parameter A, indicating the strength of the interaction between each of these pairs of genes constituting transcription factors and targets. As each target can have multiple TFs, this will explicitly be a weighted sum of expression for all regulators of that gene, which can be written for all target genes at once as this matrix multiplication of a gene-by-gene matrix A, which contains each of these weights Aij, and the expression vector X, which has dimension equal to the total number of genes. So now going back to our original goal, the solution to this ODE will give us a function of expression dependent on time. And so in order to have this sort of description of the system, all we need to do is to infer this matrix A, which contains these weight parameters for our problem. So A, based on our design, can actually be interpreted as a gene regulatory network, where each column gives the impact of a transcription factor on each of its target genes. And so this matrix will be 0 wherever there is not an interaction between a specific transcription factor and that target, and it will be either positive or negative where there is an interaction depending on whether the TF activates or represses that target. And so how do we accomplish this inference problem? So as a first step, we adopt a probabilistic formulation where now the velocities are modeled as a normal distribution centered at this matrix product, similar to what you would do in a linear regression problem. Also, as in linear regression, we have this unknown parameter A, which we need to infer. But unlike in the case of regression, our dependent variables are also latent. And so we cannot simply just find a maximum likelihood solution for A in order to solve this problem. So in this setting, where we have both a latent variable and a parameter, we will instead use the expectation maximization algorithm to generate a maximum likelihood estimate for A while marginalizing out this latent from the likelihood function. And so this sounds great, but of course, in order to do this in any meaningful way, we must constrain this problem, or else we will have non-identifiability on our solution after marginalizing out for these latents. So in order to do this, first we will adopt a biologically motivated prior on the matrix A itself, which works by first defining a binary version of the GRN, which basically describes which TFs might regulate which target genes without denoting the extent to which or the sign of the interaction representing activation or repression. To this end, we leverage ATAC-seq data, which measures chromatin accessibility genome-wide. And this is well established as a method for identifying potential transcription factor binding sites and has been used in many previous works as a basis for a GRN structure. So given these potential TF target pairs, which we get from ATAC-seq, we still do not know the magnitude of the relationship or, importantly, whether each transcription factor is activating or repressing that target. And so thus, for each of these non-zero elements, we consider the co-expression between the corresponding TF target pair in our single-cell RNA-seq data, where the correlation magnitude and sign can be thought of as a prior on the strength of the TF impact on that target's expression. All of these elements together define a probabilistic prior on A to constrain our inference problem. So on top of this, we also want to constrain our latent directly, which we reason can be accomplished by using manifold level information. So specifically for each cell, we can define regions which are spanned by its nearest neighbors in local space, and we assume that likely future fates, and therefore velocities, will point toward these dense regions, as opposed to off the manifold entirely. scKINETICS is the first algorithm, to our knowledge, to use this manifold level data as prior information constraining velocities up front in velocity inference, although it has been used in the past for post hoc refinement of velocities in methods such as cell rank. So to summarize briefly before we dive into the results, scKINETICS is an algorithm which is capable of jointly learning this gene regulatory network and these per cell velocities. It does so iteratively with the expectation maximization algorithm, and it conditions both of these values on biologically informative priors from ataxic, co-expression, and the local manifold based on the nearest neighbor graph. And so this brings us into our results, which are intended to basically ask whether or not this scKINETICS algorithm can actually recover any of this sort of underlying biological process and the sort of mechanisms that underlie this. So in this case, we're going to look at this example of pancreas regeneration, which is quite apt for this because we know that in this case we have these cell types called acinar cells, which upon an injury or an inflammatory event to the pancreas undergo a transdifferentiation to become cells which are known to be duct-like, and we term this process acinar to ductal metaplasia. So this sort of switch in cell state is the exact sort of setting in which we want to understand what are the dynamics, and in particular what are the mechanisms that are driving this sort of event. And so in order to do this, we collaborated with the Scott Lowe lab, also at Sloan Kettering, and we were able to actually model this process by taking a mouse and inducing an injury to the pancreas of that mouse, and we can collect cells at different time points following this injury. So here I'm showing what the single-cell RNA-seq data looks like coming from this, and we can see very clearly based on the gene expression patterns that we have an axis that spans from acinar-like states to duct-like states, where we're gradually down-regulating acinar genes, gradually up-regulating duct-like genes, and we can capture this pretty well using pseudotime algorithms to give us an element of sort of ground truth in this system. Beyond this, we also know that this switch, that this change between acinar and duct-like states is governed in part by the activity of several transcription factors, although each of these in previous studies has only been defined by very low throughput experiments. And so we know some that we should be looking for, but there's also opportunities here for additional discovery. And so we applied our algorithm to this data set and projected velocities in 2 dimensions so that the arrows overlaid on this embedding reveal the direction of progression predicted by scKINETICS in this case. And this clearly shows that we inferred an axis from these acinar-like cells to these duct-like cells as expected. Quantitatively, we compared the direction of each cell's velocity vectors to a set of vectors derived as ground truth based on this known axis that we get from pseudotime. And we found that scKINETICS vectors generally point from acinar-like toward duct-like states as would be expected, which would correspond with the 0-degree angle further to the right on these histograms. So you can see the distribution of angles here relative to ground truth. In this case, more accurate angles will fall closer to the 0. We compared to competing algorithms, which are based on splicing kinetics, and found that these vectors, in this case, often fell around 90-degree angles, and in these cases, this indicates that the predicted vectors are actually orthogonal to the ADM axis, and therefore are largely incorrect. Beyond just accuracy, we wanted to see whether scKINETICS improves on other features of velocity, such as the local consistency in velocity vectors between a cell and cells within its neighborhood. We hypothesized that because of our manifold constraint, neighboring cells' vectors will generally point in more similar directions along the manifold as is desirable and as is not enforced necessarily by previous algorithms. And so indeed, comparing the distance between each cell and its closest neighbors in terms of angle or cosine distance, we found that scKINETICS vectors were far more consistent than most competitors based on splicing, suggesting that, in fact, our manifold constraint and the general model of velocity that we're using here may be more robust to these sort of local neighborhood perturbations compared to previous approaches. Beyond just evaluating velocities, we wondered whether our mechanistic model of gene regulation encoded within scKINETICS might be useful for highlighting some of the important transcription factors that are guiding these transitions. And so to this end, we devised a transcription factor perturbation screen, which begins with taking the learned regulatory model from the A matrix and systematically zeroing out one column representing one TF. This simulates the impact of knockout of that TF such that it may no longer impact any of its target genes in the model of velocity. With this, we compute velocity under both the original and perturbed models and use the degree of perturbation as measured by cosine distance as a proxy for the strength of regulatory activity in each cell. Here we assume that a greater perturbation on velocity as a result of the knockdown indicates that the TF was more important or active in that cell. And we can do this for all TFs genome-wide to then understand the dynamics with respect to TF binding globally. And so going back to our example, we first asked whether the in silico perturbation screen can recover known regulators of the ADM process. Interestingly, we found that a major transcription factor represented in acinar cells, PTF1A, had low expression variability across the entire axis of ADM. And this was likely due to just a technical feature of the data in that transcription factors are often poorly captured in single-cell RNA-seq libraries across the board. Looking instead at the activity score from the scKINETICS algorithm, we instead found that we recover high PTF1A activity in acinar cells, and this declines as expected over ADM. In contrast, we find that a ductal-specific TF, SOX9, has comparable expression and activity scores, tracking with increased ductal gene expression. So in this case, our activity score can accurately capture patterns which are tracking with expression as expected, but it's also able to sort of impute cases for lowly expressed TFs, and in this case, this is based on the binding near target genes genome-wide that we were able to recover from ATAC-seq. Finally, we sought to screen for some novel regulators of ductal states by comparing two duct-like clusters, finding that while SOX9 is globally upregulated along ADM, its activation is higher in some ductal populations compared to others. In fact, zooming into the heterogeneity that we see at these ductal endpoints, we actually found that there are 3 distinct duct-like clusters, which have yet to be characterized in any previous studies. We hypothesized, based on these inferred regulators, which are both related to a stress phenotype, that one of these, this purple population here, emerges in response to increased cellular stress. Indeed, we found that these populations preferentially upregulate heat shock and translation-associated factors, which have, of course, previously been associated with cellular stress. And so this is one example of a case where scKINETICS can be used to both understand the dynamics of what's actually occurring, but also a potential mechanism guiding these dynamics over time. And so with that, I would like to acknowledge Julia Zhao, who is my co-first author on this paper, and all of the Pear Lab for all of their help and advice on this over the years. I'd like to acknowledge the Low Lab for working with us on collecting the data and performing the experiments for this acinar to ductal metaplasia sequence, and also members of the Theis Lab who we've had many useful discussions with on this over the years. And with that, I will take any questions. Thank you.